And thank you also, Pipo and Rosario, for the invitation to come back to Messina. It's always a pleasure to come to Italy and see many friends and colleagues. So following on from Gabriella, I wanted to talk about some of the new approaches that are in development for the treatment of COPD. Just to let you know, I have some conflicts of interest, but I hope it won't interfere with my presentation today. So you've already heard from Gabriella that COPD is a complex respiratory disease, and really, given the effectiveness of steroids in the treatment of other inflammatory diseases, it's perhaps surprising that steroids are not more effective in COPD. Because they're not as effective in many people with COPD, I think there's clearly a need to find new medicines for the treatment of this important condition. So I don't need to tell this audience about the burden of COPD. We know that it's a growing cause of morbidity and mortality, not just from cigarette smoke, but also from exposure to oxidant pollution in the environment. And there's a huge unmet need, particularly for treating the underlying inflammatory response, which you've heard so nicely from Gabriella earlier. And I put this slide up to remind you that we have 4, if not 5, really large clinical studies giving glucocorticosteroids for many years, which have absolutely no effect on the decline in lung function that is accelerated in patients with COPD, suggesting that although they're very powerful anti-inflammatory drugs, they're clearly not having the effect that they have in other diseases such as asthma. And that has led to a whole range of different drugs being developed for the treatment of COPD, including small molecules against many of the cytokines that you've heard about from Gabriella, a number of biologics, many of which have failed, even though they've worked in the treatment of asthma, and then the use of off-label drugs that have been often developed for the cardiovascular disease, but have actually shown to be effective in some patients with COPD. And I'll come back to this because I think there is now a growing recognition that COPD is not just a disease of the lung, but of course has many comorbidities, particularly in the cardiovascular system. And I will talk about also a few other treatments later on. One of the things here, this is my only Italian slide, Pipo, it's here for you, is that there are many, many different approaches to treating inflammation under review, one of which is bromelain, which is a natural agent and shown here to be effective when combined with N-acetylcysteine. And again, this may be an effect through one of the cytokine mechanisms important in inflammation in COPD. But I want to specifically talk in more detail about 3 drugs that I've been involved in developing. And the first one is the novel PDE3/4 inhibitor, which is the first bifunctional drug having both bronchodilation and anti-inflammatory activity in one molecule. Originally, this drug was called RPL554. It now has a name. It's called ensifentrine. And this is actually a drug that the first person to do the experiment on this drug was Dom Spina, who we celebrated this morning with a prize, who was a really good pharmacologist who worked with me over many years. But I also need to acknowledge this man, Sir David Jack. David, who died unfortunately very recently, was the man who discovered salbutamol. He also discovered salmeterol, beclomethasone, fluticasone. His team discovered ondansetron and sumatriptan. And if anyone deserved a Nobel Prize in medicine, it's this man, because he really, in the respiratory area, has helped a lot of people. And with David, we set about trying to develop a new class of drug for the treatment of respiratory diseases, particularly COPD. And just to remind you, you've heard already from Gabriella that phosphodiesterase 4 is the predominant enzyme found in inflammatory cells. And when you give roflumilast, you inhibit this enzyme and switch off inflammatory cells. So we wanted to find a drug that mimicked the effects of roflumilast, but also at the same time could inhibit phosphodiesterase 3. This is the main phosphodiesterase found in airway smooth muscle. And if we inhibit phosphodiesterase 3, we get bronchodilation. So if we inhibit both enzymes, we will end up with both bronchodilation, anti-inflammatory in one molecule. And this indeed is what we've gone on to show, and I will share some of the data with you. And if this is approved later this year by the FDA, it will be the first in this class to be of use in the treatment of COPD. Now, just some of our early experiments to show you that if we gave this drug by inhalation to primates, we could inhibit allergen and also methacholine-induced bronchoconstriction. And these experiments helped us choose the dose that we went into human beings for the first time. And we also showed that this drug was effective in comparison to fluticasone propionate and roflumilast at inhibiting inflammatory cell inflammation in the lung. And then with Mario and many people, including Dom Spina, we also took this drug through toxicology and into human beings for the first time, and we summarized some of the early clinical trials which were published in Lancet Respiratory Medicine some years ago now, showing that this drug, when you inhale it, clearly is a bronchodilator and an anti-inflammatory in a single molecule. And the very first people who had this drug gave a very profound increase in FEV1. These happened to be subjects with asthma who inhaled this drug, ensifentrine. You can see giving a very, very marked increase in FEV1 compared to placebo. So we knew very early in the development of this drug that, as we'd seen in the primates and our preclinical work, this drug is a very powerful bronchodilator. With Mario, also, we very early on showed that by inhalation, this drug also improved lung function in patients with COPD. And we did this experiment very early on to make sure that because the drug inhibited phosphodiesterase 3, which is also found in the heart, that we weren't giving any cardiovascular liability. And indeed now, as I'll show you shortly, this drug has been in more than 3,000 people, and there is almost no cardiovascular liability. So this experiment early on was very important for us to make a decision to move forward. And then with Professor Dave Singh in Manchester, we also took volunteers. They inhaled lipopolysaccharide, which induced a neutrophil infiltration into their sputum. And when we gave this drug at the same dose that causes bronchodilation, it inhibits inflammation and the recruitment of neutrophils into the lung. And we've heard from several speakers over the last 24 hours, and particularly from James Chalmers this morning, that we need drugs that specifically suppress neutrophil infiltration that are not steroids, both for COPD and for bronchiectasis. And here we have a drug that does exactly that. So my good friend Visha Vazika, who many of you all know, who's Professor of Respiratory Medicine at Imperial College in London, she wrote a very nice editorial that accompanied this paper, suggesting that if this drug worked the same way when it moved into phase 3 clinical trials, that it could be one of the most substantial advances for some time in the management of patients with chronic airway obstruction. And indeed, this is what happened, because just last year, there are 2 phase 3 clinical trials called Enhance 1 and Enhance 2 that have looked at giving ensifentrine chronically to patients with COPD, and looked in both patients taking LABA or LABA, in some cases dual therapy, and a smaller number taking triple therapy. And one of the things that is very clear, as we'd seen in the very first volunteers that we used this drug in, you can see there is a very, very marked improvement in lung function compared to placebo when you inhale ensifentrine, both at the beginning of the study and at the end of 12 weeks of treatment. But most importantly, there was a very significant reduction in exacerbations. So there was a 42% reduction in the risk of a COPD exacerbation in patients taking this drug regularly on top of other medicines over a 24-week treatment period compared to placebo. So this is quite an important effect on COPD exacerbation, and really confirms what we suspected from the beginning. The drug is a bronchodilator, an anti-inflammatory, and as a consequence, not only improves lung function, but could also reduce exacerbations. And the other things that were found in these studies, the secondary endpoints, it reduced the need for rescue medication, it improved quality of life, and very importantly, on the bottom of this slide, there were no side effects that are noted compared to placebo. Now, the discussion after Gabriella's talk about roflumilast, we know it's approved for very severe COPD, but of course it has many gastrointestinal side effects. This drug has no gastrointestinal side effects, it has no cardiovascular liability, and these two clinical trials and the earliest studies, as I say, there are now more than 3,000 people who've had this drug without any problems. So we think, and the FDA have to make a decision on this drug by the 24th of June this year, but if it's approved, it will be the first-in-class bronchodilator anti-inflammatory in a single molecule. Now, the second drug that I've been involved with is working with a company in Iceland called Epiendo, and you heard very nicely from James Chalmers this morning that azithromycin has been used beyond its antibiotic use. It has a long-term anti-inflammatory and immunomodulatory activity, but as Professor Chalmers said this morning, this is always leading to resistance after you give the drug for a year. And even though azithromycin is added to the GOLD guidelines, it is an off-label use, and actually, at the moment, it will be very good if we have an azithromycin-like compound that's not antimicrobial, so we can treat chronically to reduce exacerbations, but without the resistance. And the reason that we know, there are many studies, this is just one shown here, that azithromycin given regularly over a year will reduce exacerbations in comparison to placebo. This is not because it's working as an antibiotic, it's another pharmacological feature of macrolides. So we've developed a class of drugs called barrierides, which are actually drugs that are very effective on the epithelial barrier. And as you've heard from several speakers today, in COPD, in bronchiectasis, we know that there is epithelial barrier dysfunction. And these are drugs that can also improve the barrier function. But they're also, as I'm going to show you, really effective anti-inflammatory drugs. And these are all azithromycin analogs that lack antimicrobial activity. So I think this is actually what the field needs in a major way. And I just show you some of the preclinical data. This drug is now in phase 2 clinical trials in patients with COPD, but it's a very effective anti-inflammatory drug. It's as good as roflumilast preclinically. It's also as effective and more potent than azithromycin, but with no antimicrobial activity. That's really, really key. We've looked at this in a range of models with smoking-induced neutrophil infiltration as occurs in COPD. We've also looked at this against viral-induced inflammatory response, and in every case, it reduces the inflammatory response, even though it's not an antibiotic. It's a very, very clear anti-inflammatory agent. And so the lead compound is called EP395. We presented some data to the European Respiratory Society last year. It's been through phase 1 and is safe. It's now in phase 2 clinical trials in patients with COPD. And I hope that this drug also will be a very useful drug to add to the therapies that we have, because you can replace azithromycin, hopefully, and use azithromycin for what it was originally approved for, which is treating acute infections. So both of the drugs that the EP395 is an oral therapy, and the question is, everything that we talk about primarily has been around inhaled therapy. Why do we still need, or do we need, oral therapy? And Mario and I, many years ago, put up this cartoon, which I think many, many people have now commented on, added to over the years, that when you're exposed to cigarette smoke, you're exposed to oxygen pollution, there is inflammatory responses that spill over into the cardiovascular system. And of course, I don't have to tell the audience here about that. But if you're giving a drug orally, you're more likely to target systemic inflammation than you are by inhalation. And yet, because every drug that we've developed by inhalation is designed not to get into the blood, otherwise you have the side effects of systemic steroids, beta agonists, etc. And we've already heard from Gabriella about targeting PD4 systemically. We know that when we give roflumilast, as was shown many years ago by Klaus Rabe, also shown with the PD4 inhibitor solomilast, you can inhibit inflammation in the airway when you give a drug orally. The problem with roflumilast is it has a very narrow therapeutic window, And therefore, the dose that is approved is too low, and we cannot go higher because of systemic side effects. That is why with ensifentrine, we've developed it by inhalation, really to target inflammation but avoid the systemic exposure. So we know that treating systemic inflammation with a PDE4 inhibitor, because this drug is not a bronchodilator, reduces exacerbations in COPD. So we need to have safer orally active compounds. And one example that Mario and I have been working on with Visha Vazika, Peter Calvary and others was actually erdostane. And erdostane, like N-acetylcysteine and carbocysteine, are mucolytic drugs. They're very old drugs that people have forgotten about. But when you give this drug orally for 1 year, as we showed in this paper published in the European Respiratory Journal, it had a very clear effect on reducing exacerbations in all comers. But very importantly, the duration of the exacerbations was also reduced significantly in patients taking this on top of standard of care versus placebo. So we already have a drug in the form of erdostane that can reduce exacerbations taken orally. And the other thing that was really important is we looked at the number of exacerbations overall. There is a big reduction, and particularly if you look at the 2 post-hoc analyses that we've published recently with this, there is a much greater reduction in mild to moderate exacerbations than severe, suggesting that this could be a drug that should be used earlier to reduce exacerbations. But it argues that giving a drug orally can have a clinical impact in reducing exacerbations. Now, the final thing I want to just mention before I stop is really everything we've talked about with inflammation. The leukocytes are in blood, they have to get out into the tissue. And what we ideally need is a drug that stops leukocytes getting out into the tissue. And we now have many, many different pharmacological targets where leukocytes, eosinophils, neutrophils are involved in sticking to the endothelium, migrating into tissue. And one of the things many, many years ago, and I'm very old now so I can show this slide, I suggested that platelets did not just form clots and aggregates, as we heard about yesterday in the context of pulmonary embolism, which they clearly do. They're involved in thrombosis. But I suggested, because there was already evidence when I wrote this article, that platelets were primitive inflammatory cells, and this was a completely different mechanism of platelet activation. And the reason that I say that is if you take blood from patients with asthma or COPD, you will often find that platelets stuck to the outside of leukocytes. They cooperate. And very, very importantly, you can see here the platelets in red, the leukocytes in green. Platelets stick to the surface of the leukocytes, and they also are involved in sticking leukocytes to the endothelium. And I just wanted to show you this video, which shows you very nicely the green are leukocytes moving through the blood, and you'll see in a minute the platelets in red sticking at either side in white to these leukocytes before they migrate into the tissue. This is not thrombosis. This is an inflammatory response involving platelet-leukocyte interactions. And so if we could target this platelet-leukocyte interaction, maybe we have another form of anti-inflammatory drug, but we need to do that without causing bleeding and interfering with hemostasis. So one of the things that my group has published very recently in the Journal of Applied Physiology, if you take leukocytes, you put them in a test tube with endothelial cells, you can see very few neutrophils want to stick in the top corner there. The moment you add platelets back in, the concentration you find in blood, more leukocytes stick. And in the bottom panel is what happens if you take all the platelets away from animals. You no longer see leukocytes recruited into the tissue. So you need platelets to stick to leukocytes for them to come into the tissue optimally. The idea of simple chemotaxis that we've learned from medical school and university is a very simple concept. In vivo, it's much more complicated, and platelets sticking to leukocytes are key to producing an infiltration of leukocytes. And we've recently published this, that if you look at platelet purinergic receptors, of which there are many, and P2Y1 and P2Y12. You're very familiar with drugs that block purinergic receptors to prevent platelet aggregation, things like taclopidine, clopidogrel that are there. They do nothing to inflammation. But what we've identified is that there are ways of actually activating these receptors in a way that you do not activate thrombosis. And so I wanted to just leave you with one slide that is about to be published. It's not yet published, a drug that we've developed called MRX-2500 that completely inhibits neutrophil infiltration into the lung by blocking purinergic receptors, but it has no effect whatsoever on bleeding. So we can separate platelet activation for bleeding, platelet activation for inflammation, and we're now taking this drug forward to see if it's an anti-inflammatory drug without affecting thrombosis, because these are so-called biased agonists that they can pick which receptor activation pathway they go for, and we're very hopeful that this will be a drug that is also potentially a novel approach to treating inflammatory responses. So with that, Mr. Chairman, I'd like to thank everybody. I put this old picture up because Dom is also still working with me at this time, and to thank Mario, Gabriella, many of my colleagues in Italy, Jennifer Cricker and Mike Parnham, who've helped me on the EpiEndo project, and my colleagues at Verona Pharma who are developing ensifentrine. And you have to all hope that by June this year, the FDA say, we like this drug and it will be approved as a new therapy, as a bifunctional drug, which we've developed really as a small group of academics. We're not Glaxo, we're not a big company, we've done this ourselves, so it shows you can do it. And with that, I thank you for your attention.
Thank you, Clive. Any question from the floor? Domande? Prego, Sandro.
Thank you for your lecture. A couple of questions. How far we are from the possibility to use biological drugs in treatment? I mean, the monoclonal antibodies. Probably a lot of time, since there are, it's my opinion, less immunological pathways respect asthma. And second, you mentioned the statins, very pleiotropic drugs. So which is the potential mechanism of action in treatment in COPD?
So I think with the biologics, I mentioned just at the beginning, but there have been trials with anti-TNF and a number of other monoclonal antibodies directed against specific cytokines that, whilst anti-IL-5, anti-IG have been effective in asthma, in COPD I think we've had less success. Now maybe with the different phenotypes now, that in the eosinophilic COPD, maybe they'll have some effect. And you heard from James this morning, I think another target, one of the cathepsins, was very effective in bronchiectasis. I think targeting EPO, elastase may be quite important in COPD, but personally, why are you using monoclonals at the moment in severe asthma is because they're too expensive to use in the more wide population. And the world would quite like to have a simple tablet, because in every other parts of medicine, once a day tablets are the way to improve adherence. I don't have to tell you that we have so many different inhalers, often with the same class as a drug. It's confusing for doctors, it's confusing for pharmacists, it's confusing for patients. We need a simple approach. And also targeting one pathway in such a complex disorder, I think is an issue, because you're only targeting, unless it's absolutely core, like we have TNF in rheumatoid arthritis, but even then, some people aren't responding. I think we have to go back to looking at what I call polypharmacy. It's why steroids are so effective in many inflammatory conditions, as Gabriella said. They hit lots of things. We've become so reductionist to try and explain complex diseases. So I'm not that hopeful, actually. I know some recent monoclonals have suggested there may be some effect in COPD, but they're not going to be widely used by people because they're too expensive. So, you know, that I think is a big issue.
What about the possibility, Sandro asked you about the statin.
Yes.
What's the possibility of using inhaled statins? I have seen papers, studies performed in animals, and luckily there is some companies that is working for developing an inhaled formulation of a statin.
Yeah.
What is your opinion on this possibility?
So I think when I mention statins, there are already some suggestions and other drugs that are used for cardiovascular disease, not least beta blockers. I mean, you know, medical students are taught never to go near a beta blocker. But if you look at the meta-analysis from Scotland and other places, with beta blockers, the steroid consumption goes down, hospitalization goes down. We need some large prospective studies because we may already be sitting on an answer with drugs that have been developed somewhere else because we've lived in silos. The same with statins. So the statins, there are some trials show positive effects, some not so positive. Inhalation would be one way to get around all the muscle problems if you give it locally, but you lose the advantage of a drug that we can give orally. Because when you inhale it, you know people don't know how to use their inhalers properly. And I used erdostane as an example. Here is a drug we've had for 40 years, given for 1 year, orally, on top of standard of care, can reduce mild to moderate exacerbations by 50%. Finally, it's been added to the guidelines as add-on therapy. So why aren't we using more of it? Because frankly, the effect is real. So I think the trouble, Mario, with developing inhaled drugs is there's a whole new challenge of how is the drug compatible with the device. What we really want is to be able to take drugs that already exist orally, but actually do some proper trials to understand which particular patient subgroup they're effective for.
And last question, what about polypills in COPD?
Well, you know, Mario, my view. I'd put erdostane, a beta blocker, and maybe one other compound like an angiotensin reserpine. You put it in one tablet, it's very simple. I'm surprised nobody's done it.
Any questions?
Muna, I have one question. Thank you very much for your lecture. What about nebulized furosemide?
Yes, well, I'm old enough to remember Sebastiano Bianco, so he used to do fog challenges as well as for measuring airway hyperresponsiveness. I think, again, we don't understand why, but it was effective in asthma. I'm not so sure about if there are any studies in COPD. And I, like your father, we've done a lot of work during COVID with nebulized heparin, because I think, to me, we know heparin binds viruses. When you inhale it, it does not cause systemic anticoagulant effect. We were using it in COVID on top of systemic anticoagulants. And that is a drug that is extremely effective against, Mario and I, many years ago, did a study in COPD on top of standard of care. So I think there are things out there that we can use more of, but we have to get out of this mindset of just having LAMA, LAMA, and beta agonists, because if they were that effective for everybody, we would not have to have a meeting.